Good afternoon, ladies and gentlemen. On behalf of Christian Hansen, we very much welcome you to our first Bacillus Symposium to try to go a little deeper to find out about these good bacteria that can benefit the animal health industry and particularly the poultry industry. My name is John Dickerson, and I lead the folks that do the poultry work in North America, and we also have colleagues in South America and around the world. Along with me on my team, we have sales managers here today, Jimmy Clark, Kelly Petty, and Fernando Alaruja from Brazil. And we also have our nutritionist that works with us, poultry scientist, Dr. Herb Kling. And I know you folks know some of them way better than myself, but we all get around to see different people at different times. We're very fortunate today to have the three gentlemen that are going to speak on various aspects of the bacillus. And to introduce our first speakers, we'd like to welcome David Harrington, our Global Poultry Products Manager. David?
I'd just like to extend my thanks for all of you coming this evening. I think these kind of things are important for sharing information. Okay, you know, we live in a global world. We live in a global information. So I think these kind of things are great. We can pull together experts to share their experiences, their knowledge, and help impart that information to us. So again, thank you for coming. First off, our first speaker is Professor Simon Cutting. He's Professor of Molecular Biology at the Royal Holloway, University of London. He's been there for 9 years. He leads a research team of 9 people, and at the moment they're focusing really on Clostridium difficile, TB, and influenza. Before that, Professor Cutting was in the US for a while as a doctoral researcher at Harvard University, and then as an associate professor at Pennsylvania University. He's a leading authority on bacillus spores, and bacillus spores as vaccine vehicles and as probiotics. So it's a real pleasure that we've got him all the way over from the UK today. So, Professor Cutting, please, the floor is yours.
All right, thank you. Yes, thank you very much for the invitation and also the chance to be here in America. I've got 20 minutes, so I'll try to summarize and make this as interesting as possible for you. Bacillus and its use today in the feed and also the food industry, only the former is of interest to you. This obviously is where I work at the university, very nice building. Starting with Bacillus. Bacillus is a bacterium. It's a gram-positive bacterium. It's one of a group of organisms that forms spores. Bacterial spores are dormant life forms. They can survive for millions of years. They are heat stable. You can put them in boiling water for a short period of time. And later on I'll show you can actually put them in foods which are heated to 235 degrees. So shown here, you'll see a bacterial spore. And this is what today is used in many probiotic products, both for humans and in animal feed. Just remind you a little bit about the biology. Bacteria, if you remember from your undergraduate days, bacteria grow by binary fission. They produce 2 daughter cells, which then replicates, and the cycle resumes again and again and again. Most bacteria have this vegetative life cycle. Spore-forming bacteria are able to do something different. That is, when those bacteria starve, when they run out of nutrients, they take a different life cycle, which produces a spore, and that's shown as the blue sphere inside the cell. And that spore is released, and it will stay in a dormant, inactive form indefinitely, unless you give it nutrients, and the spore can germinate and resume normal cell growth. So these spores, bacillus spores, that's spores of members of the Bacillus genus, and of course there are many species. Those spores are used in, to my knowledge, at least 5 animal groups. Obviously, pigs, cows, and poultry, most relevant to you. 2 days ago, I was in Vietnam working or supervising groups there who use spores for feeding shrimps. That's Penaeus monodon. And in fact, spores can be used to colorize shrimps, to change the color of a shrimp. And lastly, the biggest, fastest growing industry now in Southeast Asia is catfish. And spores are being used to feed farmed catfish. One of the interesting things about bacterial spores is that for any of you, if you're sufficiently interested in what I'm saying today, and you go tomorrow and you look at your textbook or look online, it will say that bacillus is a soil organism. And the reason for that is if you go outside with a bucket and spade, take some soil, heat it up, you will recover spores in the soil. That doesn't mean that organism lives in the soil. It just means you can find spores in the soil. What we've been able to show, and in fact my group years ago was the first to show this, was that spores are shed, are deposited in the soil, which after many years accumulate. In fact, after hundreds of years, the soil is full of spores, but they're dormant, they're doing nothing. But when an animal, so here's your pig, eats a plant, and this could just as easily be a human, the plant is contaminated with spores from the soil. Those spores enter the gut of the animal, they pass through the stomach, because the spore, not only is it heat resistant, it's resistant to the stomach acids. Other bacteria are not. So if it was lactobacillus, it will start to be degraded in the stomach. But a spore is completely immune to gastric juices. The spore passes through the stomach unscathed. Once it reaches the ileum, the small intestine, this is a region which is nutrient rich. The spores germinate en masse, and we've been able to show this using molecular biology techniques. They replicate, 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 and eventually, by peristalsis, as the bacteria pass through the guts, they enter the colon, they resporulate, and they're shed in the feces. In fact, if you look in human feces, you'll find 10 to the 4 spores per gram of feces. And in fact, genomics is now telling us that 30% of bacteria in the human gut are spore-forming bacteria, not necessarily bacillus. But you can see how a bacterium can carry out its entire life cycle within the gut of the host. And in fact, all animals are consuming foods which are contaminated with soil, which are contaminated with spores. So which bacillus are in use today? Well, there are a number of bacillus, and I won't get into which strains are better than others. What I will say, I can just show you some examples here. QPS, Qualified Presumption of Safety, is something which is relevant for the food industry, and it's set by EFSA. But these strains, these species, Bacillus subtilis, Licheniformis, coagulans, and pumilus, are considered safe for human use. And there are products used in animals, and in some cases in humans. And at least in Bacillus subtilis and Bacillus coagulans, there are now strains which have fully affirmed GRAS status. And then we have a number of strains, species, which are so far unassigned as being qualified, having a qualified presumption of safety, Bacillus firmus and indicus, but they are currently being used in animals, and both of them have self-affirmed GRAS applications, which are in now, today. So how do bacillus work? What's the nature for how Bacillus probiotics work? It's always the question everyone wants to know. And there are a number of mechanisms. And I've worked with a number of these, and I would say at the top, stimulation of innate immunity is the #1 mechanism for how spores, consumption of spores, has a probiotic effect. and specifically interaction with the toll-like receptors. They're receptors on host cells, which in turn stimulate something referred to as an NF-beta-kappa pathway, which leads to the induction in humans of natural killer cells, and for mammals, cytokines such as interferon gamma. And all of these events have been documented. We also know that spores do something very unusual, which no other bacteria do. They absorb toxins. And we've been able to prove this. If you give a large number of spores to an animal which has diarrhea, the diarrhea will disappear. Unfortunately, from a commercial point of view, this is such large doses which it may not be feasible commercially to deliver that. But in an animal model, there's no doubt that spores can absorb toxins, so they sequester the diarrhea, the pathogen. And of course, there are two other mechanisms which I should mention. That's obviously the synthesis, production of antimicrobials, and lastly, the restoration of the gut microflora. So firstly, evidence for innate immunity. In one simple experiment, most of my work is working on a human and an animal C. difficile vaccine. But I can just give you some examples. If you just look in the shaded box here, there are two groups of animals. These are golden Syrian hamsters. And the first group we call naive. These are hamsters which have received nothing. The second group are hamsters which have been dosed orally with bacillus spores. After a certain period of time, we challenge those animals with pathogenic Clostridium difficile. So the animals which received nothing, in 42.5 hours exactly, they're all dead. But the animals which have been dosed with bacillus spores, they live for an extra 12 hours. And of course, you'll say, but they die anyway. That's not the point. The point is, using those spores is doing something. It's preventing the disease. So let me give you a better example. This is influenza. So now what we do is we take mice. This experiment has been done 7 times now. We take mice and we dose the mice 2 times with spores. Just not a vaccine, just bacillus spores. In this case, we do it nasally in the nose. 7 days later, we challenge those mice with bird flu. What we find is, if you just look at these 2 groups, this is a dose escalation study, so higher and higher doses. But you see these 2 groups where we've dosed with 5 times 10 to the 8 or 2 times 10 to the 9 spores give complete 100% protection against influenza. What it actually means is if we had pandemic flu or if you had swine flu, you'd give the animal spores nasally, because it would protect. And you would keep giving an animal, or you keep giving humans, spores nasally. And because it's 7 days after the dosing, this is innate immunity. There's no vaccine here. There's no adaptive immune response. And we can prove, again, using a very simple experiment, that the spore is interacting with a toll-like receptor. I don't have time to explain the nature of this experiment, only to say that it's an in vitro experiment. We're taking cells, and if we add a spore, we get a color reaction. And you only get a color reaction if a spore interacts with a toll-like receptor. So we start with these control experiments, and we take lipopolysaccharide, which stimulates a toll-like receptor, and we see induction. If we take spores, we get induction. But we can have another control. We take latex beads of exactly the same size as spores, and we get no induction. This tells us spores interact with toll-like receptors. In the case of influenza, you activate a toll-like receptor, you produce interferon gamma, which is sufficient to fully protect against influenza. Absorption to toxins. We know that the spores are negatively charged. If you take a spore, you put it into an animal, it's very acidic in the stomach, but the spore retains its negative charge. If you remember your undergraduate biochemistry, you will remember that if you take a protein, that is a toxin, and you reduce the pH, once you fall below the isoelectric points, the protein becomes positively charged. But spores remain negatively charged even to pH 2. So you have a negative spore binding to the toxin. So you can imagine if you had an animal, you keep giving it spores every day, the spores are absorbing the toxin. And we have examples here where we have different toxins. This is anthrax toxin, tetanus toxin. It's binding to the spore at low pH. We have now been able to take animals where we give them spores, we challenge them with a pathogen, we can extract from the cecum spores where the toxin is bound to the spore. It's a very simple method. Give spores to an animal, you sequester the toxin. And if you go to countries like Vietnam and China where people are using probiotics with absolutely no regulations at all, when a child gets diarrhea, they take 50 packets of probiotics, empty them into hot water and drink them. But the interesting thing is the diarrhea goes away. Obviously, you'd have regulatory problems if you did that in the West. But it's interesting observation. This is an experiment which is for the food industry. McVities biscuits, manufactured by it's a European company. They have put spores, Bacillus spores, into the biscuit mix. And if you heat the biscuit mix at 235 degrees for 8 minutes, you have a 1 log reduction in the number of bacillus in the biscuits. This illustrates one important point. If you take a spore, you can put it into feed which has to be heated to a high temperature. For example, with the spray drying, where you have a high temperature, when you make the aquaculture feeds, you have a high temperature. But spores will quite happily survive at a high temperature. Now, I'll just give you some examples where when you talk about probiotics, of course, there's another aspect, and that is what is the nutritional content of a spore. And there are some bacillus spores which produce carotenoids. These slides don't show it very well, but these lyophilized cultures, you see the orange or yellow pigmentation, which is the carotenoid, which is produced by the spore. So what is interesting about the carotenoids is because just as the spore is heat stable, the spore is gastric stable, the carotenoid produced by the spore is also stable to gastric juices. There are just a number of bacillus species which produce carotenoids. Bacillus subtilis, for example, doesn't produce a carotenoid. But I'll just give you one example. If you take purified beta-carotene and you incubate it in simulated gastric fluid, after 20 minutes, in fact, after 5 minutes, there's nothing left. And this fact is known to the industry. If you take the carotenoids extracted from the spore, or just the spores themselves, you can incubate it for 60 minutes, and there's little change to the carotenoid content. I myself was a coordinator of a large EU grant, where we then went further. We took bacillus, which produced the carotenoids, and we showed the carotenoids are made bioavailable and bioaccessible. They're actually taken up into the liver and adipose tissues. So you have a nutritional aspect. So finally, I'll just summarize by saying that bacillus spores, their defining characteristic is their heat stability. They can be incorporated into feed. Most probiotics, at least to my knowledge, you can't do this. They are commensals of the GI-tract, so they make it attractive where you can actually identify bacillus which come from a particular animal and repopulate the gut. Extensive current use in humans, animals, and in aquaculture. And there's a possibility for identifying novel, new strains, new products, for example, the carotenoid-producing spore formers. And finally, you can consider logical mechanisms for how bacillus spores can inhibit pathogens. It's a very simplistic mechanism. Innate immunity being the primary mechanism, followed by absorption of toxins in the gut.
Excellent,
Right.
excellent. Thank you very much for your time. I think what we'll do is, please. That was fascinating. Probably the thing I shouldn't take home is the fact of spores in my McVities digestives. I've just come away with the best justification ever for raiding a packet of biscuits. So I won't tell that to my girlfriend. So thank you. Our second speaker this evening is Dr. Lars Molbech. Hopefully that's okay. My Danish isn't very good. Lars is our R&D manager for our innovation group in Christian Hansen, Animal Health and Nutrition. He's in charge of the group that looks at new product development and product testing. Okay, so he's the guy that comes up with the new stuff. Before joining Christian Hansen, Lars was working at the University of Copenhagen, heading up a group of 5 researchers looking at pre- and probiotics in monogastrics. So Lars, please.
Thanks.
It's all yours.
Yes, hello everyone, and I'm also very happy to be here and to share some of our experience at Christian Hansen and how we are working with our Bacillus strains. And my title today is why are some Bacillus strains better than others as probiotics? So I will divide the presentation in 2 parts. The first one will come into some of the things also Simon mentioned about Bacillus species, the features and benefits as direct-fed microbials. And then the second part, I will go more into the details about how do we actually find the new winner strain and how we work with the strains. Just a little bit to set the scene again. So we'll talk about Bacillus as the genus, go down to species, and finally at the strain level, where we actually have our product names. And in the EU, all bacterial feed additives should be registered at strain level. And as Simon already has said, we are mainly working with Bacillus species that are on the QPS list in Europe and on the GRAS list in US. So where is it we want the mode of action? We mainly want to see the probiotics work in the small intestine, in the ceca, and down to the cloaca. So we have spores passing, and then they will germinate. Some will also pass by spores, and as we just heard, that can also be quite effective in itself. And then they will get out here in the end. And I will not go too much in details here, because Simon has already mentioned some of the ways that probiotics may work. We heard about colonization resistance, and that can both be like directly or indirectly. So that means it can actually either prevent the pathogens to adhere to the tissue, use their nutrients, for example, or can be more indirectly, for example, to stimulate the immune system, as we heard, or to improve tight junctions, or even to stabilize the bacterial flora that are already there, so we get an even better bacterial flora. Finally, they can produce and degrade metabolites, and that way improve nutrient utilization and digestibility. But in the end, all this is, of course, theory, and we can prove it in studies. What we are interested in is that we are improving the growth performance, the daily weight gain, and the feed conversion ratio. If that's not working, it's no good about the probiotics. So that's really the key, and also to improve the overall intestinal performance. So why are we using Bacillus? And again, Simon had already mentioned some of the points. Bacillus are actually having a quite big genome compared to, for example, Lactobacillus. They have from 3,000 to 6,000 genes, where normally Lactobacillus have from 1,800 to 3,100 genes. That means they have a really big toolbox. They can do a lot of different things. Then finally also, Bacillus are very robust. They are fast growers. They can reduce oxygen fast. They can tolerate bile and acid, mainly in the spore forms. And also then they have this very complex way of working. So they can go from vegetative cells to spore forms and forth and back, and they can produce many different types of enzymes, which all will improve the animal's growth. So now I will go a little bit into what are the differences when at strain level. And I will take this study that was made by Hugh and just recently published. And he looked at 17 totally or fully genome sequenced Bacillus subtilis. And what he found was that there was quite some genetic variation actually among all these different strains. And he listed then the open reading frames, which are the potential genes present. And he showed there were around 3,300 that was shared among these different, 3 different species here. But each of them also had their own specific genes, which could do different things. And among the different strains here, there are also shared genes. So this actually shows there is a potential to find the right strains that can do the things we like them to do. So how do we then find these winner strains that are doing what we like them to do? The normal way we're doing is to have a big bacterial input, and that can either come from Christian Hansen's private culture bank, where we have more than 16,000 deposits. And there are many different types of bacteria, and also we have some fungi. Or we can go out and look in the environment and find perhaps the right strains we like to have. Then it's very important, the next step, and I will come back to that, that's the phenotypic screening. We have to be very clear what it is we want to have from our strains, and then we have to test for it in order to find the right features. And if and when we find these right phenotypic traits, it's also important that we can produce the new bacterial strain in big scale, because it's no good if we cannot get it up and get a product out there. And finally, and the most important, is that we test it at the farms. So how do we do the testing? Is by high throughput phenotypic screening. And we have a department in Christian Hansen that is doing these things. And they're mainly using these robotic platform with automatic pipetting and sampling handling in order to do the assays. And we can test for many different features. And here we just listed some of them, like pH tolerance, bile resistance, antibiotic resistance, growth kinetics, and so on. So that means we are generating a lot of data. And one example of one of the data is here about enzyme production, that's cellulase. And this was a collaboration we had with Copenhagen University and has recently been published. And here we can see the different Bacillus species, amylophacians, for example, Pumilus licheniformis. And we can see there is quite some variation, both among the different species, but also inside the different species, so different strains having different features. So you can imagine now that we have this data set. We are testing all the different other things. We can test immunity as well, and so on. That means we create a huge display of data. So then how then to go on and then really find the strain we like to have in nature, or as a new product, sorry. We use complex data analysis, and that is mainly, for example, PCA, multivariate data analysis. This can look a little bit complex with this figure, but I will just show you how it works. So each dot represents one bacterial strain. So we have many strains. I think there are 260 strains tested here. Then each color represents a Bacillus species. So up here, the light blue is, for example, Bacillus amylophacians. And they are like clustering together. We have subtilis, the orange dots here. And there are some variation among the different strains inside the species. And what we have on the X and Y axis are some key variables, parameters, that we rate very high. And then you can see that if you have like all these data sets, then you can combine them in different ways and then really find the key parameters that you like to test. But of course, we have to go back to the farm. We find the strain that we think is working. And then we have to test it there, and we have to do it in meta-studies. It's not enough just to do one trial and then see it's working. We have to test it again and again under different conditions in order to know that this is actually going to be a winner. And if it's not working, then it's back again, choose another strain. But then the data we actually generated from the farm studies can also be added to this meta-analysis as well. So we actually can figure out which features is it we like to have. So what I hope to show today is that Bacillus species are superior probiotic feed additives for poultry and pigs due to their big genomes, that they have a big toolbox. They are spore producers, and they can improve the overall intestinal performance. And finally, that Bacillus strains are different. And there we have to look at both the genetic potential, their phenotypic performance, and that actually we have to prove it in the end on the farms in order to get a new product. Thank you.
Thank you, Dr. Malvik.